So good day, I'm Charlene Campos and my advisor is Sir Harrison Cabrera. I will be presenting my thesis entitled Prediction and Analysis of Antigenic Determinants in Dengue virus type 3 through Immunoinformatics approach. Dengue is one of the most common types of viral infections that can be found mostly in tropical and subtropical regions. It is transmitted by an arthropod carrier commonly known as a mosquito called Aedes aegypti, which is said to carry and transmit viral infections to humans. There are about 390 million cases of dengue infections that occur worldwide, and it is caused by a virus of the Flaviviridae family and four serotypes, namely dengue virus 1, 2, 3, and 4. Sanofi Pasteur developed Dengvaxia, which has been developed in the market for many countries such as the Philippines and Mexico to be used in clinical practices. This is the first licensed vaccine in the world for dengue prevention. Aside from the licensed Dengvaxia, there are many vaccine candidates for dengue that are now in trials. The dengue virus genome is about 11,000 bases of positive sense single-stranded RNA that codes for 3 structural proteins, while the next 7 genes encodes non-structural proteins. The E protein is responsible for a wide range of biological activities, including the binding to host cell receptors and fusion to and entry into host cells. In addition, the protein stimulates host immunity responses by inducing protective and neutralizing antibodies. Therefore, the dengue E protein is an important antigen for vaccine development and diagnostic purposes. An epitope, also known as antigenic determinant, is the part of an antigen that is recognized by the immune system, specifically by antibodies, B cells, or T cells. CD8 plus T cells, or cytotoxic T lymphocytes, are the ones to recognize the degraded fragment of the viral proteins on the surface of infected cells that are being generated by the class 1 MHC bound. They induce the release of antimicrobial cytokine for the purpose of killing microbial infected cells, and it kills infected cells directly by releasing proteolytic enzymes. Next is helper T lymphocyte. It clears the pathogens residing within and outside the cells. They produces several interleukins and supports B cell stimulation and differentiation. It also produces the Th1 cell responses and activates the B cells, cytotoxic T cells, and other cells not involved in the immune system. And last, we have the linear B lymphocytes. Antibodies play vital roles in the control, including the prevention and complete removal or destruction of both extracellular and intracellular pathogens, as well as in response to vaccination. It activates the complement cascade, it neutralizes the replicating power of viruses, it prevents the diffusion of toxins and bind to their enzymatic active sites, and it also plays inductions of the macrophage and neutrophils for the purpose of clearing the extracellular bacteria. With the LPL topes, it can also ensure the long-term immunological protection. Prediction of these epitopes is therefore key to rational vaccine prediction. The objective is to predict an epitope-based subunit vaccine consisting of cytotoxic T lymphocyte, helper T lymphocyte, and linear B lymphocyte epitopes derived from the dengue virus type 3 spike glycoprotein, haloimmunogenic adjuvant, and other necessary elements through immunoinformatics, and to predict antigenicity, allergenicity, and toxicity of the epitopes for a possible vaccine construct made from the predicted epitopes. This is the overview of the methodology. First, the FASTA sequence of the dengue virus 3 and below protein E was retrieved from the UniProt database. For CTL epitope prediction, a total of 148 MHC class 1 for the 12 supertypes were predicted using the NetCTL version 1.2. The IEDB class 1 immunogenicity tool projected 75 of them to be positive in terms of immunogenicity. The toxin spread and Allertop determined that all 75 epitopes were non-toxic and 67 of them were non-allergenic. Only 17 of the 67 immunogenic, non-toxic, and non-allergenic epitopes were successful in crossing the antigenicity threshold of 0.5 according to Vaxigen. The top 5 most antigenic epitopes were chosen for vaccine construction. For the HTL epitope prediction, within the 25 percentile rank, the IEDB MHC2 allele binding prediction identified 112 unique epitopes. Among them, 74 epitopes reached the Vaxigen antigenicity threshold of 0.5. The toxin spread server determined that all 74 antigenic epitopes were non-toxic, and only 6 of them had been considered as non-allergenic by the Allertop. After considering the IFN gamma, IL-4, and IL-10 inducibility, 5 epitopes remained that met all the requirements. For the LBL epitope selection, IBCL database predicted 74 potential epitopes. 40 of them met the Vaxigen antigenicity criterion. The toxin spread database predicted that all 40 of these epitopes were non-toxin, and the other top predicted that 28 of them were non-allergenic. The top 5 most antigenic of the 28 were chosen for vaccine construction. After the prediction and assessment of the CTL, HTL, and LBL epitopes, 495 random entries of the dengue virus 3 and below protein E sequence were acquired from the homologen database and were checked for mutations through multiple sequence alignment. The relationship of the sequences was examined using the MVU server. Calculating HLA alleles distribution among the world population is critical for effective epitope vaccine design. Therefore, worldwide population coverage analysis of the above-mentioned MHC classes 1 and 2 epitopes was done using the IEDB tool. And for the vaccine construct, the top 5 selected CTL, HTL, and LBL epitopes will be combined. The vaccine that will be constructed following the sequence, the adjuvant, CTL, HTL, and LBL epitopes. For the assessment of the vaccine construct, it includes antigenicity, allergenicity, toxicity, and physicochemical properties. A highly antigenic vaccine shall be chosen as it determines the ability of an antigen to evoke an immune response and memory cell formation. The vaccine should also be non-allergenic and non-toxic. Finally, the actual immunogenic profile of the vaccine is to be estimated. In silico immune simulation will be carried out using the CNSIM server. The parameters for the simulation are proposed to be at 3 injections, each administered 3 weeks apart. Time step points for each injections are established to be 184 and 168 respectively. So for the results, first, retrieving envelope protein E sequence for dengue virus type 3 is the initial step in antigenic peptide prediction. The UniPrep database listed about 495 amino acid sequences of the dengue virus E protein from serotype 3. Shown in Table 1 is the top 5 most antigenic using Vaxgen for MHC class 1 epitopes of the dengue virus 3 envelope protein E. We wanted the epitopes to have a high antigenicity because antigenic variation refers to a pathogen's ability to modify its surface protein such that it can circumvent the host's immunological attacks. It also involves several mechanisms, including the varying of surface protein space, shifting and drifting of surface protein antigens. Hence, the higher the antigenicity, the better the epitope. In this table is the top 5 MHC class 2 epitopes of the dengue virus 3 envelope protein E. We selected the epitopes that has a percentile score not greater than 10. The reason behind this is we wanted to have a higher binding affinity of alleles for the selected epitopes. This table is the MHC binding tool of the IEDB database that were able to predict MHC alleles in 5 CTL that have 11 allelic partners and 5 HTL lymphocytes that have 14 allelic partners. Shown in this table is the top 5 most antigenic using Vaxgen for linear B lymphocyte epitopes of the dengue virus 3 envelope protein E. So this is the MView of multiple sequence alignment viewer for the B and T cell epitopes, where one mutation was found within the 15 selected epitopes out of the 495 sequences. The LBL epitope LRK, as seen in this slide, was shown to have one mutation. It had a change of sequence, meaning it doesn't conserve, and that indicates potential loss or gain of a residue. MHC HLA allele distribution differs among diverse geographic regions and ethnic groups around the world. For the population coverage, we identified MHC class 1 binding alleles. 5 CTL epitopes were considered, where Europe showed the highest regional coverage that has 96.51%, followed by North America and Northeast Asia, East Asia, while South America and Central Africa region showed some of the leads. For MHC class 2, 5 HTL epitopes were considered and almost all of them showed the highest regional coverage. The distribution of their corresponding MHC alleles in 17 geographical regions and 101 countries found in the IEDB database covers 99.72% of the world's population. The top 5 selected T cell and B cell epitopes were used for the vaccine construction. To boost the immune response, an adjuvant was attached with the first HTL epitope with the GPG linker, while other epitopes were connected using AAY and KK linkers to preserve their independent immunogenic activities after their interaction compatibility validation. Adjuvant L7 or L12 ribosomal protein was used. It showed to be, after the vaccine construction, it showed to be antigenic using the Vaxgen and Antigen Pro and non-toxin using the toxin bread. The allergenicity prediction is a key step because most vaccines transfer the immune response to the allergic reaction by initiating immunoglobulin E and type 2 T helper cells. The survey identified the constructed vaccine as non-allergen. For the assessment of the vaccine construct, total number of amino acids was 351. The ProParam database calculated the molecular weight of the construct as 37 kilodaltons, while theoretical PI of 6.68 indicated the vaccine construct to be close to neutral in nature. The instability index was computed to be 27.79, implying that the construct would retain its stability after expression. The aliphatic index was calculated to be 87.424, indicating the construct to be thermostable. The grand average of hydrophaticity was calculated to be negative 0.137, which indicated the hydrophilic nature of the vaccine. Vaccinogen and antigen Pro both predicted the vaccine to be antigenic with scores of 0.56 and 0.72. The toxin pred revealed it to be non-toxic. Allerta predicted the vaccine to be non-allergic. Therefore, epitope-based peptide vaccine construct can be predicted as a potential vaccine candidate for dengue virus type 3. Let's move on to sim presentation of an in silico immune simulation with the construct. Shown in figure A is the immunoglobulin production in response to antigen injections, which is the black vertical line. Specific subclasses are also shown as colored peaks. The simulation showed an overall increase in immune responses following repeated exposure to the antigen. Primarily, high concentrations of immunoglobulin M compared to IgG1 and IgG2 were detected. In both the secondary and tertiary responses, typical high levels of immunoglobulin activities were evident with related antigen reduction. In figure B is the lymphocyte cell population. It indicates that emergence of immunological memory and thereby proficient immunity upon subsequent exposures of the antigen. Findings were the rise in B cells for more than a year since the beginning of infection, suggesting a possibility of developing a lasting immunity after using the vaccine. Shown in figure C is a consistent rise in T helper cell population with memory development. This also indicates that emergence of immunological memory and immunity upon the exposure of the antigen. And in figure D is the concentration of cytokines and interleukins. The higher B and T cell activity and lasting of B cell memory for several days indicated humoral immunity, which is essential for complementing the immune responses. High levels of IFN gamma and IL-2 production during the repeated exposure indicates cell-mediated immune response. For cytokine IFN gamma being involved in B cell proliferation and Ig isotype switching, it can support a humoral response as well. And moderate levels of IL-10 and IL-4 activity were observed since IL-10 and IL-4 positive HTL epitopes were selected and incorporated into the vaccine construct. So this profile of the vaccine suggests immune memory development and thereby natural immune protection against the dengue virus type 3. Conclusion, this study provided valuable insights for the identification of peptide vaccine against the dengue virus type 3 through immunoinformatics. Based on this strategy, the T cell and B cell epitopes in the enveloped glycoprotein E were mapped and selected as vaccine targets. The predicted epitopes exhibited T cell and B cell selectivity, higher conservancy, non-allergenicity, non-toxicity, higher population coverage, and significant interaction with MHC class 1 allele with good affinity. Based on the parameters, this design can be considered a plausible candidate for a vaccine against the dengue virus. So, thank you for listening.